Assalamualaikum. Today I'll talk about alternative pathway of complement system activation. There are three pathway of complement system activation. One is classic pathway, two is alternative pathway, and the third is lectin pathway. In previous video I have discussed about classic pathway, and in this video I'll talk about alternative pathway of complement system activation. Alternative pathway induced by the presence of bacterial endotoxin, which is a lipopolysaccharide and present on the surface of the gram-negative bacteria. And other substances like yeast cell wall and cobra venom. The alternative pathway was discovered next to the classic pathway, and alternative pathway is the response of innate immune system. In contrast to the classic pathway, which is a response of adaptive immunity. So, let's go through the in details of alternative pathway. The alternative pathway starts from component protein 3. Component protein 3 is the most abundant complement protein in the blood. First of all, complement protein 3 undergo spontaneous hydrolysis in plasma. It forms two fragments, C3b and C3a. C3b is the larger fragment and C3a is the smaller fragment. Now, the C3b is the main component of alternative pathway. C3 is called anaphylatoxin. Because C3a activate mast cell, and from mast cell it releases histamine, and histamine causes smooth muscle contraction. And increase vascular permeability. And these are the main cause of inflammation. And now, suppose it is a gram-negative bacteria covered by lipopolysaccharide or endotoxin. And here come to the C3b. C3b come over here, and another factor called B, it is also a complement factor, come over here. Now, with the help of factor D, which is another type of factor, with the help of factor D, the factor B become fragmented. Now it become B, B and BA. The BB is the larger fragment and BA is the smaller fragment. The BA is go away from here and BB bind to the C3b. Now C3b and BB act as a C3 converters and act on C3 and divide it to fragments C3b and C3a. C3a will go away and C3b will attach to the remaining fragment C3b, BB and C3b. Now they act as C5 converters. Again, hydrolyze or fragment C5 into C5b and C5a. C5 go away from here and C5 will bind. Then C5 produce C6b, C7b, C8b and then C9b. And these are the membrane attack complex or MAC, which will attack the membrane of bacteria or other substances and making pore in the surface or membrane. Then the ion and water will enter into the cytoplasm and it will undergo swelling and ultimately the cell will die. Then another thing can happen. The C3b acts as an opsonizing agent. It can bind to the surface of the antigen and block the pathogenicity. And ultimately the phagocytic cell like neutrophil and macrophage engulf them and ultimately the bacteria or antigen will disappear. So this is all about the alternative pathway of complement system activation.